Hello friends, welcome once again. Today I am here with Indian Horticulture magazine, November-December volume 2019. So this is a special volume on potato. So coming to the first question, Global Potato Conclave was held in Gandhinagar, Gujarat, 28 to 31 January 2020. So coming to the next. Now the first question is development of advanced hybrid, that is LBY15 and LBY17. This variety have combined resistant to late blight and potato virus Y, which is developed through MAB, that is marker assisted breeding. So question may be, which of the following variety of potato developed through MAB, that is marker assisted breeding? So remember LBY15 and LBY17. Now next one is identified potato genotype having multiple resistant gene for late blight resistant and cyst nematode resistant. So just remember the name of the gene for late blight, that is R1 and R3, and for potato virus Y, that is RYADG, and for cyst nematode, that is HC, H1 and GROW1. These genes are identified through molecular markers. So just remember the name of the genes that is responsible for certain resistant to certain diseases. Now coming to next, developed transgenic potato with improved trait, durable, resistant to late blight. Earlier what the thing is, earlier transgenic is most focused on insect resistant. So in potato, first transgenic was developed for Colorado beetle, but nowadays there's more focus on late blight resistant, cold induction sweetness, and high protein contents and so on. So now new technique, that is Indo blight cast, was developed for forecasting a potato late blight. And another one is development of a decision supporting system. Just remember the full form of CAASPS, computer aided advisory system for potato crop scheduling. Now another point is India utilize only 7.5% of the total production for processing, as against 30 to 67% in leading potato processing nations such as USA, Canada and Europe. So we utilize only 7.5% of total production for processing. And now next important point is the processing genotypes should possess high dry matter more than 20%. This is important question, many time asked in exams, like maximum dry matter content more than 20%, and reducing sugar less than 0.15% is acceptable for chips and french fries. Now coming to the next, low glucosinolate content less than 15 mg per 100 gram fresh tuber weight is the standard. And now coming to the next, see, there is some interesting fact, like what kind of chips varieties earlier we are having and now we are having. And some of the varieties like Kufri Chandramukhi, Kufri Jyoti, Kufri Laukar, these varieties were used by the chipping industry from produce of crop grown in warmer area where night temperature are relatively high, around 10 degree. It's a news for when before they are using, but what is changed now? In 1990, research was going on, and in 1998 first two processing varieties, that is Kufri Chipsona 1 and Kufri Chipsona 2, were released in India. Okay, next is in 2006 Kufri Chipsona 3 was developed with higher proportion of defect free processing grade tubers. Before they are using only those varieties which are not suitable, but they are making them suitable. But now these days these varieties are suitable, especially if they are developed for chips making only. How it was possible, just see. Now some question also, just see the some questions. Processing variety suitable for hills. See the name of that variety, that is Kufri Himsona, which is suitable for hills and developed in 2008. Now next is, these are just 120 days long duration varieties, medium to long duration varieties, and now they are focusing for region specific varieties. Just see region specific variety Kufri Chipsona 4, developed in 2010, suitable for Karnataka, West Bengal and MP. And now they are focusing on French fries as demand of French fries. So Kufri Chipsona 1, which one is indigenous variety, uh, it's tuber producing low French fries grade. So see, coming to the next. Now keeping in view the long felt demand of French fries varieties, so Kufri Friesona for North Indian plains in 2009 they have developed. Before they are developed for Karnataka, West Bengal and Madhya Pradesh, now for North Indian plains. So Kufri Friesona for North Indian plains, and new variety is Kufri Frio M released in 2018, also suitable for French fries. Kufri Frio M. So if you want more information about Kufri Chipsona 1, just go through it, and then Kufri Chipsona 2, and just see the varieties Kufri Chipsona 1, 2, 3, 4, Kufri Himsona, Kufri Friesona. These are the indigenous varieties suitable for processing. Now coming to the next, this is very important question. Please note down this question. Dominant gene, that is RB gene. This is gene which is isolated from bulbocastenum, and it's a source for late blight resistant in potato. So this is a very important question. Now these days RB gene is mostly used. So the source is Solanum bulbocastenum. So just remember the name of gene and the species for late blight especially. As we know that like genome sequencing was done in 2011, so if you want, you just remember the name of the line. And another one is identification of STSP6A gene. This gene is what is the advantage of this genome sequencing? So by this you will get, scientists we will get like each gene what kind of functions they are having, like they will get all the information of each genes which is responsible for certain character. So see, STSP6A, this is for tuber initiation, and STCDF, this is for plant maturity. Now coming to the next, inter-specific potato hybrids. If you want, you just go through this C13 and this one. Now this is a very important question, just remember this. Solanum tuberosum dihaploid C13 and wild potato species Solanum pinnatisectum. Okay, Solanum pinnatisectum, Solanum cordiophyllum. These varieties for these species for late blight resistant, and E tuberosum for potato virus Y. But more specifically, you must have to remember like RB gene from bulbocastenum. Now coming to the next, Solanum freja. It's a diploid potato which is rich in ascorbic acid. So maybe it will ask in exam. So it's very important, like Solanum freja. Freja is diploid and source of ascorbic acid. And Solanum gaurlei, this is for highest beta carotene content. Now coming to the next, worldwide example of biofortified potato, that is which is developed through conventional breeding, is INIA 321 cowsay for high iron and zinc content. So these are the other varieties, just remember it. Now actually before they for late blight resistant, R1, R2, R3 gene are using from Solanum dimesum, but what was the problem? That one is narrow resistant, but for broad spectrum broad resistant, RB gene is now they are using from bulbocastenum. So please remember it. I am saying again, RB is resistant source from Solanum bulbocastenum for late blight resistant, kindly remember it. Now coming to the next, potato virus Y resistant. So the name of the gene is Rhisto gene from Solanum stoloniferum. RYAD gene from Solanum tuberosum indigenum group, and RYCHC from Solanum chacoense. These are now they are introducing into elite cultivar which is resistant to potato virus Y. So remember the name of the gene and their source. There are 2-3 species and different genes, please remember. Make a just these important questions may be asked in matching, so kindly remember it. Okay, so you just, if you want, you just go through this table: frost resistant, aphid, wort, potato virus Y, late blight. Solanum freja, it has contributed different tuber color and flavor, so it's important forager. And others are also here mentioned, just if you want you can go through this. See, linep, a cultivar developed using gene from Solanum chaquense, is in the pedigree of many modern chips cultivar, but there is some problem. What is the problem? It was removed from the market due to excess level of glycosinolate content. Hmm. So those variety which is coming from Solan chaqueans having excess glucosinolate content, that's why they are removed it from the market. And for chips, now what kind of species they are using for chips making? See, Solanum tergiens and Solanum raffinifolium, especially in cold storage. Now just remember this, if you want interspecific potato hybrid, it's important because maybe in this year now pattern is changed, so you have to go in detail or in depth, like interspecific, why they are using interspecific potato hybrid through somatic hybridization. So important question is to break the barriers, zygotic barriers, these things. So just remember these questions and their importance, their properties, what is the properties for somatic hybrids, why they are using. See, in this case protoplast fusion overcome sexually incompatible by protoplast fusion tetraploid like common potato confirmed by flow cytometry. What is flow cytometry? How we can get information like that this species is tetraploid or not? It's just a common instrumental tool for rapid detection or characterization of cells. This is based on their light scattering and fluorescence properties. So flow cytometry is that only. And then what is the properties of this protoplast fusion? Maybe they are very high resistant to lead blight resistant, having diverse genetic ways, and then nuclear and cytoplasmic genes of two solanum species in a single one, long tuber dormancy, male and female fertile, crossable with potato cultivated potato varieties. Please remember all these things whatever mentioned here, and don't think like Kripa Shankar is telling all these things in depth. Please go through this because now pattern is changed, no one knows like from where question will ask. So please, if you are like these things, so please share and subscribe. Thank you.